Hello, grade 9. How are you today? We are going today to explain about reproduction. I'm going to talk a little bit about the life cycle. I'm not going to go in deep details in it, but a general idea in order to know a little bit about reproduction. And then I'll enter in the chromosomes that are the most important issue today. So reproduction can occur in animals, either these animals human or human being or animal creatures, or plants. So reproduction can occur in both species. Let's go in details in reproduction. What does the word reproduction mean? Re means again. Produce means to make something new. So reproduction is to make something new again. Reproduction can be of two types. We have sexual reproduction and asexual reproduction. In sexual reproduction, as you can see in this figure, we should have a male and a female that mate together in order to give a new individual. However, in asexual reproduction, there is no necessity for a male and a female to occur together in order to give us a new individual. New individuals appear by just dividing the parent. When the parent divides, it will give two identical individuals as it was. So this one is asexual reproduction, where no male or female mate in order to give a new individual. Let's now compare a little bit between sexual and asexual reproduction so that we can go in details in sexual reproduction and understand it well. What I want you to concentrate on is sexual reproduction, but in order to understand the sexual reproduction, we have to make a comparison, a small comparison between these two types of reproduction. Sexual reproduction, as we said, that it needs the mating of a male and a female in order to produce a new individual. However, asexual reproduction produces a new individual without mating of a male and a female. In sexual reproduction, it includes the presence of a sperm and an ovum. A sperm is coming from the male, it's the male gamete, and the ovum is the female gamete, and these we will talk about them in the second slide. However, in asexual reproduction, it doesn't include the presence of these gametes. In sexual reproduction, we have three processes that goes on in order to have a new individual. These processes are meiosis, fertilization, and mitosis. We are going to talk in details about meiosis and mitosis later on, but now I want you to know that in sexual reproduction, we should have these three processes in order to have a new individual. However, in asexual reproduction, it is only mitosis process that occurs in order to have a new individual. In sexual reproduction, the new individual produced gives new characters. So the new individual that we have has his own characters. These characters are 50% of them from his mother and 50% of them from his father. So if I'm talking about you, you are not identical to your mother or identical to your father, but you have a 50% characters from your mom and 50% characters from your dad that you've mixed together to get you. You have your own personality, your own characters. So this is sexual reproduction where each individual has his new characteristics. However, in asexual reproduction, the produced individuals or the coming offsprings are identical to their parents, even in their genes. They are genetically identical. So even their DNA is identical to that of their parents. Let's go zoom in now to sexual reproduction and concentrate on these points that we should concentrate on them. Sexual reproduction. So it is the mating of a male and a female in order to produce a new individual. So here we have a female, and this is the female egg that we call it an ovum. Here we have the male, and the male gives the sperm cells. Okay, these are the gametes of the male, the sperm cells. When they unite together, and the process is called fertilization, where the ovum and the sperm unite together to give us a zygote. This zygote develops and grows by a process called mitosis to become an embryo. These details we are going to talk about them later on, the processes, okay? But now, a general idea that we have a female and a male. The female has a gamete, and this gamete is an ovum or an egg. The male has sperm. When the ovum and the sperm unite together by a process called fertilization, they give us a zygote that develops and they grow in order to become a new individual that is called embryo. Let's recall something. So, what I'm interested in is to go from this introduction about reproduction into the core of my interest. So I am interested in the life cycle because it leads to explain about the chromosomes. So what are chromosomes and where they are found? Let's recall a little bit about the cells. So if I took, for example, a cell from the mother or a cell from the father, or if I took a cell from the zygote, a cell from the embryo, or a cell from a plant, what do I have? So if I look at a normal cell, taking from an animal cell or a plant cell, we can see that the cells are made of many various organelles inside it. But the main thing is that in both cells, animal and plant cell, we have the nucleus that contains the genetic material that we call it DNA. So this is the nucleus, and this is the genetic material that we call it DNA. The DNA, this structure holds the chromosomes inside it, okay? So let's go inside this structure. What is DNA? What is the genetic material? What are the chromosomes? First of all, why do we need to know about the DNA? So I'm talking about the reproduction, giving new individuals. Why I need to talk about DNA? Of course, we need to know about DNA because DNA is the genetic information, the code that gives us this individual. So if I don't have this code, I will not have a new individual. So to know the identity of the individual, to know who is this individual, we have to know his DNA. Each individual has his own genetic material. To know the gender of the individual, to know if this is a male or a female, we have to study the DNA. So we have to study the chromosomes. In order to know if this individual is a normal one or abnormal one, we have to study his DNA or her DNA. So we have to study the chromosomes, abnormality or anomaly, to know if he has any abnormality. Okay, so we have to study the DNA, the chromosomes. If we look inside the nucleus of the cell, we will see the DNA. The DNA is a long strand. When it condenses, it will give us a structure called chromosome. So it is a thread-like structure spread inside the nucleus of the cell. But when it condenses, it gives a structure that's called chromosomes. So when it is spread inside the cell, inside the nucleus of the cell, it is called DNA, chromatin material. This material, when it condenses, it will give us a chromosome. Let's skip some slides and let me show you when this material is a chromatin and when it is a chromosome. If we see here, our cells divide to give many cells. We will talk about this process later on, but as a general idea, when our cells need to become more, to proliferate, this means the cells need to divide. And there is a process for this division. It is called mitosis. I will talk about it later on. But look, when the cell is normal and it is not dividing, this is the DNA inside the nucleus of the cell. It is a thread-like structure. It's not condensed out of each other. It is free. So we call it a chromatin material. However, when the cell starts to divide, this material condenses and becomes smaller in size. It is not spread anymore inside the nucleus. It is not free inside the nucleus. So it will become a chromosome structure that is found here. So how can I look into these chromosomes? How can I see these chromosomes? Of course, we cannot see cells, so we cannot see the DNA, so we cannot see the chromosomes by our naked eye. But there is a process called karyotyping. Karyotyping leads to give us and allow us to see the chromosomes inside the nucleus. Here is one of the procedures done in order to make a karyotype. So a 5 milliliter of venous blood is taken from this individual. So we took from an individual 5 milliliter of blood. We know that blood is made up of red blood cells and white blood cells. Of course, we don't need the red blood cells because they are enucleated. And the enucleated cells don't have a nucleus, so they don't have the DNA. So we don't need the red blood cells, but what we are interested in are the white blood cells. So we put these white blood cells in a culture medium. We let them to divide at 37 degrees Celsius and become more in number for 3 days. After that, we put a substance called colchicine and we let the cells to fix. What does this mean? So in this culture medium, I let the cells to divide to become more in number, and this is the case where one cell will give us 2 cells, 2 cells will give us 4 cells, okay, and so on till the cells become great in number. Then we put a substance called colchicine to stop the division of the cells. So when I have to stop the division of the cells and why I have to fix the cells and stop their division, look carefully. If the cell is in the beginning of its division, look at this material. The DNA in this way, I cannot see it very well. Look at the second stage. In the second stage, the cell is still very small. Although the DNA look in a structure of chromosomes and it is condensed, but the cell is so small, so the DNA is still very close to each other. However, the biggest cell among these ones is this cell at the stage of metaphase. So colchicine fixes the cells at the stage of metaphase that allow us to see the chromosomes very well under the microscope. So the chromosomes inside the nucleus at this stage are well seen under the microscope. So scientists put colchicine to fix the cells at this stage so that we can see these chromosomes. And then they add a saline solution, a hypotonic solution, in order to burst the nucleus. When the nucleus bursts, all the chromosomes will be spread out of the nucleus. They will be free to see them clearly. And this is what happens. So we put colchicine, we fix the cells, and then we spread the cells on a slide to put it under the microscope. Then we put a stain, coloring, in order to see these chromosomes because they don't have color, of course. We put coloring in order to see them. When we see them under the microscope, we take a picture and then we will arrange these chromosomes in order so that we can see if there is any anomaly, or we can see if this person is a normal person. We can see the gender of this individual, if he is a male or if she is a female, okay? So this is the karyotyping procedure where we put colchicine in order to fix the cells at metaphase, and then we stain these chromosomes in order to see them, and karyotype is obtained. How can I arrange these chromosomes? How could I know that this is 1 and this is 2 and this is 3? How could I know these things? Chromosomal arrangement is done according to the placement of the centromere. I didn't know what's the meaning of the centromere. Look here carefully. The chromosomes are made up of 2 sister chromatids. Can you see this structure? This one is a chromosome. Chromosomes are made up of 2 sister chromatids attached to each other by a centromere. So this one is a centromere. This one is a centromere where the 2 chromatids are attached to each other. 2 sister chromatids are attached to each other. So when I see that this chromosome, its centromere is here, and this chromosome, its centromere is also at the same level of this one. So these are homologous chromosomes. We call these chromosomes homologous because they are the same, with the same placement of the centromere. We arrange the chromosomes according to their size. The largest one first, and then the small one. So we arrange the chromosomes according to their size. We arrange the chromosomes according to their banding pattern. What is the meaning of banding pattern? If you look here, you can see bands. These bands are genes that code for something in our body. It may code for our earlobe if it is free or attached. It may code for our blood type. It may code for the shape of our nose, the shape of our hands. So these are genes. If you look at these 2 sister chromatids, we can see that the bands are at the same level and they are for the same gene. Here also the bands are of the same level. So we arrange the chromosomes according to these 3 criteria: placement of centromere, size from the largest to the smallest, and according to the banding pattern. Remember that the chromosome is made up of 2 sister chromatids attached to each other by a centromere. Okay, and we have homologous chromosomes. Let's continue. So, the karyotype will be obtained according to the type of the cell taken. So, if I took a cell from the skin, and if I took the cell of a gamete, will the karyotypes be the same? Of course, no. Let's see what is the difference between a karyotype of a somatic cell and a karyotype for a gamete cell. We have 2 types of gamete cells. As we said before, we have an ovum and we have a sperm. The somatic cell and the gamete cell. Let's see the difference. This is a karyotype for a somatic cell of a male. When I say a male has chromosomes, the sex chromosomes or the gonosomes are X and Y. The autosomes that are from pair 1 to pair 22 are the same for male and female. We are recalling this information. So from pair 1 to pair 22, these are autosomes and they are the same in males and females. But what differs a male from a female is that the gonosomes, the sex chromosomes of a male hold a Y chromosome, however that of a female have 2 copies of X chromosome. So this is a karyotype, a normal karyotype, having 46 chromosomes, 22 pairs of autosomes, and the 23rd pair is a gonosome for a male. Look at these chromosomes. we have pairs of chromosomes. So this is chromosome number 1, this chromosome is not 1 prime, 2 prime. So we have pairs of chromosomes. In this female also we have pairs of chromosomes, 1 prime, 2 prime. So we have 22 pairs of autosomes. Let's take a look on a gamete cell. This is a gamete cell. This is a sperm. Look at it. Do we have pairs of chromosomes? No, we don't have pairs of chromosomes. We have chromatids. So these are chromatids. And look here, we have only a Y chromatid. We don't have X, we have only Y chromatid. So this is a sperm of a male. It is chromatids. We call them haploid cell. However, the ones that are pairs, like these ones, we call them diploid, as if they are doubled. Diploid. Here they are haploid chromatids. We also have from 1 till 22. Of course, the chromosomes are not affected. We are talking about normal ones. But these are chromatids. However, these are chromosomes. This is the one for a female. Look, from 1 till 22. We are talking about chromatids. This is an X. The gonosome is an X, so it is an ovum. Of course, when I am telling you this information, all the karyotypes that I've used are normal karyotypes. There's no abnormalities. Now we are going to enter to the case where the karyotypes are not normal ones. Let's see together. What are the abnormalities in a karyotype? The karyotype may have an abnormality in a whole chromosome or in a part of a chromosome. If I'm talking about a whole chromosome, so I'm talking about trisomy or monosomy. If I'm talking about a part of chromosome, I'm talking about translocation or deletion. Let's zoom in each one of these structures. If the case is trisomy, what do we have? What do we mean by trisomy? Tri means 3. So this is the case where the karyotype has an extra chromosome. So one of its chromosomes has an extra one. So if I'm talking about a somatic cell, and this somatic cell has a diploid, so we have 2 copies of each chromosomes. The chromosomes are found in pairs. The case of a trisomy, that the chromosome is not found in pairs, it is 3 copies of a chromosome. So let's see together. Here we have 3 copies of chromosome number 21. So this is called trisomy 21. Its name is Down syndrome. Here we have trisomy 13. It is Pateau syndrome. Here we have a trisomy in the gonosomes, in the sex chromosome. We have 2 X and 1 Y, and this is called Klinefelter syndrome. This is the trisomy where we have an extra copy of a chromosome. This extra copy can be in the autosomes or in the gonosomes. What is monosomy? Monosomy is the deletion of one of the chromosomes. We have one chromosome that is absent. Let's see here. All the autosomes, if you look at them, they are found in pairs. They are diploid. When we come here to the gonosomes, we can see that we have only an X chromosome. However, the Y chromosome is not found. So the Y chromosome here is deleted. This is monosomy, where the karyotype has an absence of one of its chromosomes. So we call it monosomy. What do we mean by translocation? Location means place, and translocation, so something is located but not in its place. Let's see this karyotype. We have pairs of chromosomes here. So we are talking about a somatic cell, diploid cell, where the chromosomes are found in pairs till chromosome 13. Oh, chromosome 14. Why chromosome 14 has this something on the top? Let's continue. 15, 16, 17, 18, 19, 20, 21, 22, so on. The others are correct. But here, chromosome 14 has an extra copy of chromosome number 21 on it. So this is a translocation. Chromosome 21, we have an extra copy of chromosome 21 found on the top of chromosome 14. So chromosome 21 is placed on chromosome 14. So this is translocation, when one of the chromosomes is translocated on the other one. Let's see, what do we have here? We have deletion of a part of the chromosome. So all the chromosomes are of the same size except chromosome 5, where one of the chromosome, its upper part is deleted. This is deletion of a part of a chromosome. So not the whole chromosome is absent. If the whole chromosome is absent, this means monosomy. But if a part of chromosome is absent, we call it deletion. So we have 4 types of abnormalities. We have trisomy, monosomy, deletion, and translocation. This is a video to recall about the life cycle and about the chromosomes. Just take the notes if you want, and watch the second video that talk about exercises and how can we solve such exercises related to chromosomes and karyotyping. Thank you.